Welcome everyone. Thank you for joining us for our presentation titled Polygenic Risk Score Analysis for Alzheimer's Disease Risk. It's my great pleasure to introduce Dr. Richard Pither. Richard is the CEO of Cytox, a precision medicine company based in the UK. And Cytox is developing and commercializing a new diagnostic technology for assessing Alzheimer's disease risk. For a complete biography of Richard, please visit the tab at the top of your screen. This talk was recorded some months ago, but Dr. Pither is with us live today for the Q&A to provide an update and answer your questions. Please do submit your questions via the Q&A box. A reminder that this presentation is educational and you can obtain free continuing education credits. To get those credits, please click on the tab in the top right corner of the presentation window and follow the process. And now, over to the presentation.
Thank you. Hello everyone, it's a pleasure to be able to talk to you today about polygenic risk score analysis for Alzheimer's disease risk and the opportunities that this affords, much needed opportunities for transforming the prospects for development of new Alzheimer therapeutics. During the course of my presentation, I'm going to be covering an introduction to the clinical challenges of Alzheimer's disease and associated drug development. I'm going to be talking about polygenic risk scores and how they can be used, and specifically how they pertain to the genetics of Alzheimer's disease, and what we've been developing at Cytox, which is a polygenic risk score algorithm with the trade name of genoSCORE, and how that performs in longitudinal clinical studies, and the subsequent implications for stratifying subjects into clinical trials for the future, and the prospects for clinical trial enrichment. And of course, that in turn will influence very positively, we think, the prospects for new drug development. You can see on this slide, which describes the challenges of Alzheimer's disease, the huge problem that the industry faces. Alzheimer's disease is the most common form of dementia, but it is one of 4 or 5 recognized types, constitutes between 50 up to 70% of dementia. And the problem that the world faces today is this escalating problem associated with an aging demographic in the developed world. The costs today are estimated at about 800 billion worldwide, and they will treble by 2050. And one of the challenges associated with the development of new drugs is addressing the failure rate. We have not seen any significant new drug approvals since 2003, and that's associated with the problems of finding the correct subjects for enrollment at the earliest stages of the disease, where the drugs that are in development have the best prospects for working, to the point where the screening failure rate is over 90% in some studies. What I'm going to talk about today is how genetics and polygenic risk scores in particular can help improve that success rate. So of course, Alzheimer's disease is not purely a genetic disease, and I'm talking about the late onset form of the disease, as I'll describe in a moment, but actually manifests itself as a consequence of genetic inheritance and also exposure to environmental and lifestyle risks. And that's illustrated on this slide here. We're all born with an inherited level of genetic risk. The question is whether that risk becomes manifest at the level of disease symptoms. And as we can see, as we get older, those symptoms can be very mild in the preclinical or prodromal phase of the disease, and then manifest as clear dementia later on. Whether or not a defined genetic risk becomes manifest at the level of symptoms seems to be a measure of how well we manage our exposure to other risk factors, and I've listed some of them on this slide, such as cardiovascular risk, hypertension, whether we take regular exercise and eat a sensible diet, whether we have diabetes or whether diabetes is under control. A whole series of things that can be managed in a high genetic risk individual might well push back the time at which symptoms become apparent. And that's very important in terms of clinical management considerations going forward. Now, there are a number of ways we can intervene to decide whether or not someone is in a high-risk category today by the use of very invasive and very expensive biomarkers, but these have significant limitations. So if we look at the left-hand side of the slide here, we can see two curves. The yellow curve associated with normal age-related memory loss. As we all get older, our cognitive functions tend to decline naturally, but very slowly. Well, some of us will be on that more precipitous blue and red curve associated with Alzheimer's disease, and this is not a normal aging process. What is happening at the time of those symptoms, or actually about 20 to 30 years before those symptoms, is a build-up of underlying pathology in the brain associated with amyloid plaque, neurofibrillary tangles, and other pathologies. And so we have a window, we have a 20 to 25-year window where we could intervene therapeutically to actually affect the outcome of that disease process. So on the right-hand side of this slide, on slide 5, we've illustrated here a couple of the techniques that are used today in the form of amyloid positron emission tomography imaging, which is effective at picking up amyloid in brains of subjects, even pre-symptomatic subjects, but is very expensive, at sort of $5,000 plus per scan. And of course, the earlier you go in the disease process, the less likely you are to find someone with amyloid. And so typically in clinical trial recruitment, you may have to carry out 10 scans to find one positive individual. The other option here is a lumbar puncture, where we're taking out a sample of cerebrospinal fluid to look for telltale amyloid and tau protein fragments. But that's actually quite difficult to do quantitatively and reproducibly across many labs. And of course, it's highly invasive, having to take a lumbar puncture sample. So we've been working on the premise really set out by Professor John Hardy at UCL some years ago, that genetic analysis would be the most cost-effective way of deciding who should be assessed using these more invasive techniques for early signs of disease development. On this slide, you can see actually that there are two forms of Alzheimer's disease, and our focus today is on the late onset form, by far the most common form of Alzheimer's disease. Probably upwards of 95%, maybe as much as 98% is associated with late onset, by which we mean greater than 65 years of age. There is an early onset form, of course, which occurs in families, in discrete families, and these are associated with dominant mutations in the amyloid precursor protein and the 2-presenilin gene. These are very well documented. Late onset disease, on the other hand, is associated with ApoE locus, as we'll talk about shortly, but also with a large number of loci throughout the genome. These 21 loci here were documented a few years ago in a well-known publication, but since then there have been a much larger number identified, and these have small effects, and it's the cumulative effect of these variants that seem to confer risk for the late onset form of the disease. So Alzheimer's disease, the late onset variety, is associated with many DNA variants spread throughout the genome. As of this publication, back in 2018, there were over 40 different loci identified, each of which the variants confer either a little bit of added risk or perhaps a little bit of protection for the development of the disease. You can see on the right-hand side here, the ApoE locus is by far the most commonly associated with increased risk. But depending on where you draw the cutoff in this so-called Manhattan plot, we can identify large numbers of other risk-associated DNA variants. So, to summarize what's needed on this slide, we can see that we need effective early identification, ideally at the pre-symptomatic phase, of at-risk individuals. And the solution that we're talking about today is a polygenic risk score approach, and our particular algorithm we call genoSCORE, which is driven from a DNA sample, either blood or a mouth swab, which is then isolated from that sample, the DNA, and run on a proprietary array that we call variaTECT. We'll talk about that in a moment. That variaTECT array is built on a Thermo Fisher Axiom platform, run on the gene Titan equipment. And then the output from that array is analyzed in our software platform, a proprietary platform called SNPfitR, within which the genoSCORE polygenic risk score algorithm sits. So we get the primary genotyping data from the array that's analyzed using genoSCORE in an automated workflow run in whichever lab we're working with that happens to have this equipment. So we work regularly with Axiom in the US, with Eurofins in Europe, and we've worked extensively with the Ramachotty Centre in Australia. But essentially, we can work with any lab that has this laboratory infrastructure. Okay, so the variaTECT 2 array will be familiar to some people. We previously had the variaTECT 1 array. This is a new version which contains something like 5 or 6 times as many SNPs, including all the original variaTECT 1 content, plus optimized SNP panels to drive the polygenic risk score algorithms. Now, the discussion today is really all about the genoSCORE algorithm, but just for information, we do have other polygenic risk score analyses that we can carry out from the basic genotyping content of this array, and that analysis would be available depending on exactly what the experiment was trying to achieve. But you can see here, we've got various panels of SNPs, some of which arose from our collaborations at University College London with John Hardy and his group, many of which come from Cardiff University and are optimized to drive the algorithm on which genoSCORE is based. We'll talk about that shortly. And some of them come from mTOR-associated pathways, which are from University of Birmingham collaboration in the UK, which allows us to drive other forms of analysis. We have an extensive literature panel covering all the early onset associated variants in APP, PSEN1 and PSEN2. And of course, we have panels that would allow us to do pharmacogenetics, ADME type studies, as well as imputation if that was required. So the Cardiff algorithm on which genoSCORE is based is one of the best published algorithms that's available, and it has some very key characteristics. It was trained in a very large database, a very large training set, the so-called IGAP consortium data set, and contains a very large number of SNPs. So we're talking here about an algorithm which, in this publication, the best accuracy was achieved using more than 87,500 SNPs or DNA variants. Actually, the genoSCORE algorithm that I'm describing now has got a bit more than that. It's got a few more, 115,000 or so. And we find that this gives us the most stable prediction across different data sets relating to Alzheimer's disease risk in the samples that we've analyzed, and for which I'll show you some data shortly. A final point on this slide is that the validation of the Cardiff algorithm has been done in completely independent data sets, some of which are pathology confirmed, which is a kind of gold standard for Alzheimer's disease definition. And so we know that the performance is not just due to the overfitting in a training set, but indeed is preserved in completely independent test sets, which is a very important characteristic. And you can see some data here from exactly those sorts of test sets, which we'll cover in a bit more detail on the subsequent slides. So, of course, the Cardiff algorithm is not the only polygenic risk score algorithm, and I would draw your attention to this paper that was published last year by Hannah Stocker et al., which set out to compare the performance of different polygenic risk score algorithms for predicting Alzheimer's disease. And essentially what Hannah and her colleagues concluded was that of the algorithms available, the Cardiff algorithm is the best performing, the highest performing in terms of accuracy, and it's one of only 2 that have been actually tested and confirmed in pathology confirmed data sets. So that's very important when dealing with a disease which can be quite difficult to accurately clinically diagnose without that post-mortem histopathology correlation. Now, one of the key characteristics we're going to look for in any polygenic risk score algorithm is how it compares to assessing risk above and beyond ApoE4. You'll recall that I mentioned a couple of minutes ago that ApoE4 is a risk-associated allele. In fact, it's the most characterized of the risk associated to alleles in for late onset Alzheimer's disease. But carrying E4 itself may give you an elevated risk, but does not in any way determine whether or not an individual will develop Alzheimer's disease. So it's important if you are an E4 carrier, and this slide illustrates the distribution in a general cognitively normal population, in this case from the UK. So about 23% of the population carry one copy of E4, the so-called E3, E4 heterozygotes, and these would be considered to be at relatively high risk, but we know that a lot of those people will not go on to develop Alzheimer's disease. Conversely, the majority of people will not carry E4, but will fit into the E3 homozygote group, and yet we know something like 40 to 50% of Alzheimer cases come from that E3 group. So we can see automatically here that E4 assessment alone will not be sufficient to have any clinical utility in predicting the future likelihood of developing Alzheimer's disease, and indeed that's the case. Now, just before I get on to share some data, a little point here that our job really in Cytox has been to take and build on the academic excellence of the Cardiff research algorithm and turn it into a validated product that can be then used ultimately in the clinic. So for that, we've worked across a number of different data sets, including ABLE in Australia, University of Pennsylvania, Emory University, and others. And we have had to demonstrate robustness in all ApoE genotypes. The algorithm I'm focusing on today comes from Cardiff University, but there are others available too that do slightly different things. For example, the algorithm from the University of Birmingham that we've developed based on the mTOR pathway. We can talk about that on another occasion, or people can ask questions about that should they wish. The array is a customized array built on the Thermo Fisher Axiom array platform for reasons of quality control, essentially. And we've got global access via our partners, Pither, Eurofins, and other laboratories. So if we begin to think about the data now, why would we want to use, how would we expect to be able to use this type of approach? I think there are several key reasons. I mean, one, as I've touched on already, is to identify the clinical trial participants at the earliest stages who are most likely to cognitively decline during the follow-up period of a therapeutic trial. So this will allow us, of course, to reduce trial subjects and the associated costs and increase the chances of demonstrating a therapeutic effect. We said already that we would need to demonstrate performance above and beyond that associated with ApoE genotyping alone. We'll talk about that shortly. And of course, what would be ideal would be able to predict those at highest risk of meaningful cognitive decline, by which I mean objectively measurable cognitive decline within the period of a clinical trial follow-up. And then another point on this slide, the last point actually, is to do with subtypes. So the expectation is that some drugs are more likely to be effective in some subsets of Alzheimer's disease sufferers than others. And this type of analysis lends itself to being able to effectively identify those genetic subgroups and therefore enrich in subsequent therapeutic studies in a way akin really more to cancer drug development and the use of prognostic markers. The work I'm going to describe now has all been analyzed using the so-called ADNI data set. ADNI stands for the Alzheimer's Disease Neuroimaging Initiative. It's a long-running study that started in 2004 and comes with very comprehensive longitudinal follow-up data. And the subjects have been confirmed using PET imaging. So that gives us a high level of confidence based on the amyloid burden in these subjects that the clinically defined Alzheimer cases really are Alzheimer's disease rather than some other dementia, and that the age-matched controls really are controls and not simply pre-symptomatic at-risk Alzheimer's subjects. So most of the work I'm going to be describing now has been done in this cohort. There are over 630 amyloid-confirmed subjects, and these are very well age-matched across controls and subject groups, a mean age of about 78. And what we can do in these analyses is compare the performance of our algorithm with other forms of algorithm based on ApoE alone or combinations of ApoE and other parameters, as I'll describe shortly. So if we look across the ADNI data set and we ask how good are we, how good is genoSCORE at predicting clinical AD from cognitively normal controls across all ApoE genotypes, in other words, this is everyone thrown in together, we can see that if you do that using the ApoE alone, you have an AUC, an accuracy measure essentially of about 73%. The Polygenic Risk Score algorithm without ApoE component gives you about 70% in an ApoE independent measure. And if you combine the two things together, we come up with an AUC of 80%. So that's a significant improvement above ApoE. But of course, this is in subjects who, some of whom will be ApoE4 carriers. So if we ask the question, what happens if we look at the E4 high-risk subset carriers within this group? So these are the heterozygote E3, E4 carriers. Are we able to differentiate relatively high and relatively low risk in this group? We can see that by establishing cutoffs using our polygenic risk score analysis, in this case, the top quartile fell at the 0.61 and above cutoff. We have a positive predictive value of 84% at the AUC level. So that's telling us we're able to pick out the top quartile, the highest risk subjects amongst that E4 carrier group, and these are the people you might well want to enrich your clinical trial with. And actually, if you take that measure down, that cutoff down, to give you the top 50%, you're still at 71% positive predictive value. And this would be hugely valuable to anyone wanting to enrich a study cohort for those most likely to develop the disease, and therefore the ones that should be recruited into your clinical study. If we take the other end of the spectrum, the ApoE3 homozygotes, who would be conventionally thought of as having relatively lower risk, we can see here that we're able to stratify, in this case by taking the lower quartile with a negative predictive value of about 89 or 90%. So we definitely want to eliminate those subjects from the clinical trial. And on the other hand, if you took the highest quartile, you'd be up at about 50%. So positive predictive value of about 51% in this particular study. So you might want to include some of these in your clinical trial so that your indication would extend to those E3 homozygotes within the study parameters, within the inclusion-exclusion criteria for your study. So if we look at the conclusions from the work I've just described and from other studies that we are continuing to carry out right now, we can say that the stratification of the E4 carrier heterozygotes can identify the subset of E4 carriers at the highest risk of developing Alzheimer's disease. And of course, these are the ones you would want to stratify and enrich into your clinical trial to measure therapeutic effect. At the other end of the risk spectrum, the ApoE3 homozygotes, our analysis has clearly shown it's possible to identify the subset of these individuals at the lowest end of the risk spectrum, and would also allow you to identify the higher risk E3 homozygotes to include in your study design as well. The algorithm has the potential to improve the ability to predict individuals who will convert, therefore, from mild cognitive impairment to AD well over and above ApoE alone. And given the observations that enrichment of the MCI and the other prodromal at-risk subjects gives you the highest opportunity to show therapeutic effect, this is a very important observation. And we're doing more work on this very question at the moment using the ADNI cohort and indeed using other cohorts as well. The algorithm clearly has the potential to select those patients who are most likely to decline cognitively within the time frame of a clinical trial. And that's obviously crucial, because without that decline in a reasonable period of time, therapeutic effects are very difficult to measure. It's worth mentioning that our genoSCORE and indeed other algorithms are available for research collaboration at this stage, and we're working with a number of pharma, biotech and academic studies at the present time. And going forward, our intention is to be engaging with the US and European regulatory authorities and look for full marketing approval for clinical version of our genoSCORE algorithm in the 2021 timeframe. I want to finish by acknowledging our team and other partners, very important partners, a very talented team here at Cytox in the UK, and we work globally with collaborators around the world. Key partners for us, as I've mentioned en route, have been the Dementia Research Institute team at Cardiff University, led by Julie Williams and Valentina Escott-Price, and in particular, two very talented postdocs, Effie Ballo and Ghana Lianenko, and of course John Hardy at the UCL Dementia Research Institute, and Mariam Shoai, again, John's postdoc there. We've, of course, relied heavily in this presentation on the ADNI data sets. I'm very indebted to ADNI and, of course, Mike Weiner, the PI for ADNI, and not least all the patients, volunteers and their families who contribute very important samples and, of course, their time to commit to the extensive clinical evaluations that are very important in participating in those sorts of studies. Our funding at Cytox comes from a number of sources, not least our investors, but also from grant money. We've received, continue to receive money from Innovate UK, and we also receive money from European Union. So thanks very much for your attention, and I look forward to answering your questions in due course.
Thank you very much, Richard. A really great presentation on a very hot topic, polygenic risk scores, their potential for future clinical applications, and a really, really important public health challenge as well of dementia and Alzheimer's. So it's my real pleasure now to welcome Dr. Richard Pither for our live Q&A. As a reminder, please do submit your questions via the Q&A box. We've got a good number already in, and we will get through as many as we can. So, Richard, this talk was recorded some months ago, actually. Would you like to provide us with an update on where the Cytox story is today?
Yes, of course. Nick, I just checked that you can hear me.
I can.
Okay, good. Yes. Well, actually, we've made quite considerable progress. We are launching genoSCORE, not just as an academic and pharma research tool, but as an approved laboratory-developed test through clear accredited laboratory partners in the US and in Europe for clinical use later this year. So this is a huge step forward for us, and we've held several clinical advisory board meetings with various European and US partners, and actually have another one tomorrow with Australian partners. And there's huge interest in clinicians being able to access this type of polygenic risk assessment in order to help them diagnose and then optimize the treatment and care of their own clinical patients and subjects. So that's a very exciting move for us. And actually, some of this has really been motivated, I think, by the current COVID pandemic and situation around the world. The fact that we can run our test from DNA extracted from a simple saliva or mouth swab sample is a huge advantage in the sense that a kit can be sent to a subject in their own home. Some of their cognitive function can be assessed remotely, and then that sample sent back to a lab for analysis without the patient having to even leave their own home, let alone present themselves, in a clinical environment, a hospital or a primary physician facility. And thereby reducing the risk of exposure to COVID in the process. So I think that's been a sort of unexpected benefit, really, in this COVID time. So we're progressing with that aspect of it very quickly. And probably the other element that we've managed to make progress on, Nick, in the last few months since we recorded this presentation is the correlation of polygenic risk score of our genoSCORE result with underlying biomarker data. So I mentioned earlier in the presentation that the kind of gold standard measures of Alzheimer risk today involve looking inside the brain directly or indirectly, either via imaging, PET imaging, or via lumbar puncture and CSF testing, looking for telltale accumulation of amyloid and tau protein. We've done lots of correlation work recently, which actually has just been accepted today for peer-reviewed publication, where we show that those with the highest polygenic risk score are the ones who are building up these amyloid and tau fragments. And yet we can make this assessment without the need to have to do these very invasive and very expensive tests.
So there's some really tremendous progress in the Cytox story there. Thank you for giving us that update. And we questions coming in from our audience as well. So we'll fit a few of those in just now, if we may. So one of the questions is, how has your polygenic risk score been validated? So has it been verified in an independent way?
Yes, absolutely. I mean, the original training of your algorithm was carried out by our colleagues and collaborators at Cardiff University using the so-called IGAP consortium data, and there's 50 to 60,000 clinically defined Alzheimer cases and age-matched controls in that data set, and that was used to develop the algorithm. Then, of course, what you need to do is test the performance in independent data sets. The Cardiff team did that initially with the academic version of the algorithm in data sets such as the GERAD data set, and also in post-mortem histopathology confirmed cases and controls, the kind of gold standard data set, including one called the TGen data set, which is an Eric Ryman Banner Institute data set, and others as well. And then what we've done is that we've taken the Cardiff algorithm, we've worked and developed the parameters, we fixed the parameters, and then use that to prospectively analyze performance in independent data sets. And a very important one that I mentioned in this presentation was the so-called ADNI data set, the Alzheimer's disease neuroimaging study that's been run for the last 15 years or more by Professor Mike Weiner at UCSF. And so that's how we validated the clinical performance to date. We're also continuing to work with partners, academic partners, on biomarker and other sort of data sets, a longitudinal data set, and that will also make up part of our formal clinical validation data that we'll be presenting to the FDA in the next 12 months or so.
Fantastic, thank you. So we'll move on to our next question from the audience as well. So the question is, can the same gene score be used in other disorders? And maybe it's helpful to just kind of describe who you think might use what Cytox has developed as well in the frame of this question.
Okay, so what we're looking at here is specifically an algorithm, genoSCORE, that's been developed for predicting future development of Alzheimer's disease. And of course, that can be further refined to identifying those at highest risk of cognitive decline due to Alzheimer's disease. And that's important because cognitive decline is one of the key measures against which efficacy of any intervention, be that a new drug development in a trial, or be that some other non-drug based intervention in the clinical management of a patient, is assessed. So what you need to be able to do is measure cognition at baseline and then cognition at some future point. And it's the change in that cognition that then gives you some confidence that that intervention is having the desired effect. So that's critical for us, and that's critical for physicians and their patients' health. But of course, it's also critical for pharmaceutical companies whose clinical trials are looking to enrich study population at highest risk of decline due to Alzheimer's disease. But key to that is getting people at the very earliest stages of the disease, when clinical symptoms alone are not necessarily enough to accurately forecast those who will decline cognitively. So that's really the key population that we're positioning our test for. And as I said, we're getting lots of interest from pharma and from clinicians alike. Mm-hmm. Now, could this be used in other diseases or indeed other forms of dementia is a critical question. We've done a little bit of work in other dementia types, and but we're not making any claims about performance in those dementias. I mean, polygenic risk score algorithms generally were developed initially actually in schizophrenia, but not this algorithm. This algorithm specifically has been and is being validated for performance in Alzheimer's disease-associated dementia. So that's quite an important differentiation. Perhaps the other point I should make is that, as with any genetic test, you need to define your population. And so the development and the validation of performance has been done almost exclusively in Caucasian subjects, for the simple reason that's where the high quality research GWAS data, genome-wide association data, has been derived, and that's where the well clinically characterized cohorts actually exist. Now, we have ambitions to move beyond Caucasian populations, but again, we're making no claims today about performance outside the Caucasian group at this stage.
Thank you very much indeed. That takes us a little over time. We do have a number of other questions that have come in. I will come to those in a moment because there is an opportunity to speak further with Dr. Pither. So first of all, let me thank you, Richard, for the great presentation and the update. Thank you also to you guys, the audience, for the great questions that have come in. Now, two opportunities to have those questions answered if they've been submitted today and we haven't got to them. So firstly, they will be addressed by email after the event. But secondly, if you would like to speak further with Dr. Pither, he will move over to the networking lounge now, and I will host that chat session. And you can join him there to ask questions and interact further. Now, if you haven't been to the networking lounge yet, you can just click on the icon at the bottom of your screen. And when you get to that part of the virtual conference, you will see Dr. Pither's name listed on the agenda list. And if you click his name, that will allow you to very easily enter the chat room. Just a couple of other points as well as we wrap up. This presentation will be available for on-demand viewing after the event. So please do share it with a colleague who might be interested in polygenic risk scores and today's topic. I'll now close this presentation. Thank you. We hope to meet you in the expert lounge shortly. Until then, goodbye.